All right, guys. So, immune system stuff. We're going to start off by talking about your non-specific or innate immune response. Innate meaning what? Built in, born with it, instinctual almost. And by far the most important part of that innate or built-in part of your immune system is your skin. Your skin is a barrier, obviously, kind of like your Great Wall of China. But in addition to being a barrier, it also makes chemicals. For example, sweat has what waste product in it? Urea, which is toxic, and that kills germs. Your skin also gives off acidic secretions that kill germs and antibacterial secretions. So, skin keeps out germs, keeps in what? Yeah, but mostly, what are we concerned about keeping in? Water. Because between your skin and your organs is a whole layer of muscle. So the skin's not really about keeping in your organs. It's about keeping in water. Then in addition to skin, you've got inflammation. Anytime tissues are damaged, whether it's burn, cut, scratch, whatever it may be, you have cells in those damaged areas called mast cells, and they release signals, basically like SOS signals. And the main one of those is histamine. And what histamine tells your body is essentially, hey, I'm hurt over here, send more what? Blood. So the blood vessels in the area of the damage dilate, get bigger, more blood comes to the area where the damage is at. So you get more platelets, you get more white blood cells to heal up the wound, to clot the wound, and so on. In addition, your body has this protein in it called interferon. What does interferon interfere with? The lytic cycle, and that's the what? Yeah, virus reproduction cycle, where they attach, inject their DNA, reproduce, put together, burst out, essentially. Your stomach acid is also part of your non-specific immune system. What's the pH like inside your stomach? 2-ish. What acid is the main acid? The hydrochloric, same stuff you used in chem lab. Sometimes we might call it gastric acid in biology. Gastric just means what? It's in the stomach. It's HCl. It's hydrochloric. Pretty much anything that goes in there dies. We've got your normal flora. Now, normally, flora means what? Flower, plants. But here it means what? The bacteria and other microorganisms living in your body. And remember, we said that the flora sort of acts sort of like an offensive line for you. That there are these good bacteria that are living there. So what can't happen if the good ones are there? Other things can't grow there. It's like they're blocking for you. They're on your skin. They're in your digestive system. They're in the female reproductive system, blocking out other things from growing there. Yeah.
So like a risk of being on antibacterials is killing those and having other stuff in the bin.
Yep. That's why a lot of people, when they're on antibiotics, can, especially women, get yeast infections. They can get diarrhea, vomiting, because you're killing off the good stuff and the bad stuff. All right. You've also got saliva, and we could also throw in tears here. Saliva and tears have an enzyme in there called lysozyme. What do we know lyse means? Break, cut, tear, split. And that breaks, cuts, and tears and splits germs. That's why you don't get eye infections very often. You don't get infections inside your mouth all that often. Because saliva and tears have materials in there to get rid of germs. Your mucous membranes, part of your non-specific immune system. They filter out germs, so they don't make it inside. Your mucous membranes also do what else that's really non-immune related? What else are they important for, other than just filtering? They warm up the air when it's really cold, and they do what else to the air when it's really cold? They moisten it. Because think about how non-humid it is when it's really cold. If you're out there breathing that, your lungs would dry up. Don't want that to happen. Your mucous membranes moisten for you on the way. So that's just some of the more important parts of the non-specific part. In general, usually they're going to ask you about the specific part of your immune system. So that's what we're going to focus on today. The specific part is sometimes called acquired immunity because you have to build it up. You have to learn it. You have to acquire it. What is an antigen? That's a key word. Foreign protein. And in response to antigens, well, that's what our immune system responds to, right? We can either make antibodies against those antigens, or we can attack cells that are displaying those antigens, our own cells that are displaying. So let's go to this diagram and remind ourselves of some things here first. We'll start with the diagram on this side first. This purple cell is kind of a non-specific immune response piece. What are these big cells like this big purple one? Macrophages. And they just eat anything in your body that's not supposed to be there. They're big eaters, macrophages. And once they eat something, they put it in a vesicle, they phagocytize it, put it in a vesicle, and then what organelle comes over and dumps in some enzymes? Lysosome. And what process happens to that thing? Intracellular digestion. It's broken into pieces. These little triangles represent those pieces, and we call those what? Antigens. And this big green thing, we're going to call what? MHC2. Only like phagocytic cells in your body have that. And the job of that protein is to do what? It does antigen presentation, which essentially means that it's going to take those antigens from that thing that this cell ate and digested, and it's going to hold them up on the outside. And it's essentially saying, hey, this thing was in me, I killed it, here's what it looks like. I need you to do what? Go kill more. And who's it telling that to? This green cell down here at the bottom would represent your helper T cell. Helper T. Because the helper T cell's job is then to do what? We compared it to what historical figure? Paul Revere. Okay? So the macrophage is going to activate the helper T cell, but only a helper T cell that's specific to what? That particular antigen. And a lot of these are going to clone after they get activated. And then they're going to send out messages to two other kinds of cells. And the messages that they're going to send out are called interleukins. Or sometimes you might hear them called cytokines. Either way, interleukin means what? Between white blood cells. And what does cytokine mean? What's it telling the cells to do? It's telling cells that are specific to that antigen to reproduce. So cytokine here, you should think what? Cytokinesis is telling those cells to divide, make more. And the two kinds of cells it's talking to are mainly what kinds of cells? B cells to make what? What do B cells make? Antibodies. And what's the other kind of cell? The killer T cells, which go and kill your own cells that are what? That are infected. And it's stimulating both of those, sending out the warning. All right, and then over on this side, this just represents a somatic cell. It's a normal old body cell. And it's got an MHC on it too, and it's called an MHC class 1. And what is it displaying? It's displaying proteins from inside itself, which would normally be what kind of proteins? Normal proteins, good ones. But occasionally, if it's got a virus inside it or some kind of other germ inside it, maybe it's mutated and become cancerous, the antigens that end up here are not normal. And what cell is going to come along and realize that? Yeah, either a helper T, and it's going to send out more messages, or eventually killer T cells specific to that antigen will dock to this MHC, and then what do they do to this cell? If it's infected, they essentially stab it. I compared it to going Wolverine on them, right? They stab it, kill it, it dies, so that that cell can't spread the infection. And that's where we're going on this next slide. Thought it was the next slide. Well, I lost my picture. It's there, according to this. That's really weird. You guys see that the picture's showing up here, but not there?
What's that sign?
It says this image cannot currently be displayed. All right, let me.
Close the document.
All right, so back to what we were saying. Two parts of your specific immune response. Antibody-mediated immunity, sometimes called humoral immunity, because it can only get to the germs that they're where? In the body's fluids. That's what humors are. Because it attacks with antibodies, and what are antibodies made out of? Big proteins that are bulky and charged and can't get where? Into cells. And then you've got cell-mediated immunity, which kills your own cells that are sick. Let's talk about the antibody side first. So you got an antigen. The first time this antigen has ever been in your body, first time you've ever been exposed to it, it gets engulfed by a macrophage. Intracellular digestion happens. It displays the antigens on its MHC class II and stimulates the helper T. The helper T then sends out its interleukins or cytokines and stimulates B cells. But only B cells that are what? Specific to that antigen. And essentially what the helper T cell is telling those B cells is, number one, you better do what? Reproduce fast. Clone yourselves. So the B cells specific to that antigen start cloning themselves. Most of those B cells eventually transform themselves into what are called plasma cells. They're called that because they have a whole lot of what organelle? ER, rough ER. Which means they're doing what if they got a lot of rough ER? Making a lot of proteins and then doing what with those proteins? Sending them out. And those proteins that they are making and sending out are what proteins? Antibodies. Antibodies specific to whatever that antigen is, right? So they start doing that, make thousands of them per second, just over and over again. The rest of the B cells become memory cells. So they know what the antigen is, they know how to make antibodies against it, but right now they're just waiting. When the infection's over after a few days, what happens to these plasma cells? They die, they do, what's it called? Apoptosis. They're programmed to self-destruct. They go away. How long are these memory cells there? Probably the rest of your life, at least many, many years. And we'll come back to memory cells in a minute. There are also memory helper T cells that are formed. So these two together help to give you long-lasting immunity against that germ. We'll talk about how that happens in a second. At the same time, you've got cell-mediated immunity going on, specifically if this is a virus infection. So the virus, infected cells are displaying these antigens, these virus antigens. Your cytotoxic T cells will go around and recognize those cells displaying those virus antigens. They'll be stimulated to clone themselves too, the cytotoxic T cells. Only the ones specific to those antigens are cloned. Most of them become active cytotoxic T cells, killers. But they'll only kill what? Cells that are displaying that particular thing. A few of them hang around as memory T cells. So these go out, they go Wolverine on all your sick cells, stab them, kill them, make them go away. The infection's over. You've got memory cytotoxic T cells, you've got memory B cells, you've got memory helper T cells. Together, both of these parts of the immune system are specific, and without both parts, you probably would never get over a virus, right? This gets the viruses when they're in your fluids, this gets them when they're in your cells. You got to have both parts to get rid of it. And like we said in here before, how does HIV work a number on this?
Attacks.
Yeah, HIV wipes out your killer T cells, so that essentially this never gets told to start working, and neither does this. So essentially, what are you left with? in terms of your immune system?
Your innate.
Yeah, you're left with the stuff on the first page, the innate stuff. You're down to skin and inflammation and stomach acid, which is not all that effective. That's your specific immune response, and this is what part of your specific immune response. If it's the first exposure, what do we call this? The primary immune response. So with the primary immune response, let me get to the graph that I want here. Primary immune response, here we're looking at the amount of antibodies in your blood plasma, the liquid part of your blood, against time. So here's the first time you're exposed to a germ, or in this case a vaccine. Works the same way. How long does it take your specific immune system to actually get going good? 5-7 days. So your antibody levels start to go up, and they get higher and higher and higher, until eventually by the time they get to right here, probably what's already happened when they max out. You're probably over it. And so then what starts happening to all the B cells and plasma cells, they start dying. And what starts happening to those antibody levels? They start dropping. And they drop down to close to zero. But what's still floating around in your blood?
The memory cells.
The memory cells are still there. So here, this is 60 days later, here you're exposed to the second dose of the vaccine, or maybe the same germ gets in your body again. So that happens right here. And then this is called the secondary immune response. And notice that it makes way more antibodies than the first time around, and it makes them how? Way, way faster. Instead of 5 to 7 days, it happens almost immediately. So that the second time around, if this were a virus, what would happen? You probably would never get any symptoms. You wouldn't even know you were sick. Your antibody levels would shoot up. They would kill the thing. Your cytotoxic T cells would start killing any infected cells very quickly, and you would not even notice any symptoms. So this is primary immune response. It's pretty small, kind of slow. After it's over, though, there's memory cells. Memory B cells, memory helper T, memory cytotoxic T. So that the next time around, the response is much, much faster, much, much bigger. Probably don't ever get sick. A couple other things that we need to look back over. Here's a picture of an antibody. An antibody is a protein. And notice that in these pieces of the protein, that looks almost like what? Those little parts of the protein. Well, it's like a groove. So it's like an active site. And what has to fit in that little active site? The antigen, okay, the antigen that it's going to attack, right? So antibodies are a lot like enzymes. They're very, very what? Specific. And that's why this is the specific immune response. An antibody against this year's flu probably not going to help you against next year's flu, right? Because what happens in a year with the flu virus? It changes a lot, it mutates, and the proteins change, so it won't fit. Point being, this part, this active site, or what they're calling up here the antigen binding site, that's the part that has to attach to the antigen, and it's really, really, really specific. Now, we also mentioned that we only have a handful of genes to code for antibodies. So, with only a handful of genes, how can we make antibodies against all the different things that we have to make antibodies against? Remember that our genes, since we're eukaryotes, have introns in them. Those are the non-coding parts. And exons, coding parts. And let's just draw a diagram up here. Actually, never mind. I'm not doing a diagram right now. What's treated as an intron in one case might be treated as what in another case? An exon. So what that allows, each one of your genes is really what? Many genes. And what protein actually is made depends on what? What your body needs at that point in time. So like I said, we only have a handful of genes to code for antibodies, but because of alternative DNA splicing and RNA processing, we can actually make millions of different antibodies. because those few genes can be arranged in different ways. They can be recombined to make different proteins. And then, another thing or two. Let's talk about what antibodies actually do. Typically, the antibodies actually kill things. Not usually. Antibodies can do several things. One thing they do is they bind to a germ. Let's say that Sean here is a germ. And let's think of this stapler as like a flashing light. This is my antibody. And I'm the antibody, and hold your hand there, Sean. It attaches to Sean, and he's got it held up like this. So he's a bad guy, and now he's got a flashing beacon on him. And yeah, just imagine the bright black flash coming out of that stapler there. And what's that flash going to attract? What do we want to actually kill him? What are the killers? The macrophages are the actual killers. But think of them like big, dumb, slow, hitmen. Okay, they're not real bright. But if you say, kill him, they're really good at that. Okay? And that's what that antibody is doing. It's saying, kill him, he's the bad guy. And they go and kill him. So it makes the process of the macrophages eating the germs much faster. Another thing they can do is they can attach to an antibody and be almost like a handle. This time Lucas is the bad guy. Antibody attached to him. Alright. I'm a white blood cell. Lucas is dangerous. This antibody is telling me Lucas is dangerous. I mean, look at it. Doesn't he look dangerous? It's kind of scary. So as a white blood cell, I don't want to touch him. So this antibody acts like a handle. And I grab the handle. And I drag him. Come on, I'm dragging him. Where am I dragging him to? Nope. I'm dragging him to the kidney. Okay? And then I let him go. And what's the kidney going to do? He's going to end up in the toilet because it's going to filter him out of the blood, and I'm going to urinate him out, and he's gone. All right?
I thought you were the white horse.
Well, I'm mixing analogies here, but you get the idea. Okay?
No, I don't. You're being very
The white blood cells will take the germs to the kidneys, which will then flush them out, and they'll be urinated out of your body. That's why if you have a virus infection, what will the doctor tell you to do? Drink plenty of fluids. Drink your fluids so you can urinate those germs out.
So that analogy, like, what was the antibody doing?
The antibody basically just attaches to the germ, and it acts like a handle for the white blood cell to grab it and drag it.
So the antibody gets flushed out.
Yes.
Okay.
All right. Let's see if there's anything else here I want to mention on this for right now. All right, we'll mention this, and then we'll switch gears and we'll do a little statistics here. We talk about different types of immunity. If an immune response is natural and active, what just happened? If you have a natural active immune response, you got sick, you made antibodies, you fought it off, and now you've got what? Memory cells. You're immune to that germ. It's the old-fashioned way. Now, on the other hand, if it's natural and passive, what happened? Yeah. You were a baby and you nourished your mother, and what did you get in the breast milk? Antibodies, which protected you. Or you're still in your mother, and you got antibodies across what?
The placenta.
Across the placenta. Either way, you have antibodies in you that you yourself didn't make. So that's why it's passive, natural passive. If you're having an artificial active immune response, what's going on?
Vaccination.
Yeah, you got vaccinated. And what did they shoot into you? Yeah, they either shot in dead virus, which is really a stupid term, right? Because viruses aren't really alive in the first place, but inactivated viruses might be a better way of saying it. Or maybe just what? Maybe just pieces of a virus. Sometimes they might not inject the whole thing. They might just inject pieces, antigens from it. But does your body know the difference? No. So you still got foreign proteins. So what do you make?
Antibodies.
You make antibodies and memory cells. And it's like you had it anyway. And when the real one gets into you, the first time you're going to have what response? The secondary immune response. You're not ever going to really get sick. All right, and then if you have artificial passive immunity, that's when what's going on? Yeah, you're getting antibodies from another person, either through like a blood transfusion, like they did with some of those Ebola patients, or they actually have, like if a doctor gets a needle stick from a patient, they have these injections called immunoglobulins, which are essentially just antibodies that they will give the doctor to keep the doctor from getting sick. Sometimes it works, sometimes it might not.
Does that also go under like the third immunity or whatever, like when one person can't get a vaccination for some reason and then most other people do?
Right, it protects everybody.
Yeah.
I mean, that one person's still probably not going to get sick because most everybody else has been vaccinated. All right. So, questions there? All right. Does everybody have a formula sheet, an AP formula sheet? If you got one, get it out. If not, Matthew, look and see if there's some of those laying over there.